Hello everyone, in this video we are going to learn how many fragments will be generated if you cut a linear DNA and circular DNA with the same restriction enzymes. Okay, the problem is echo R1 enzyme recognizes the sequence GAATTC. Okay, echo R1 is a restriction enzyme which is used to cut a DNA double helix, and it recognizes the sequence of GAATTC and it make a cut in between GA, okay, at 5' end. Okay, so the first question, a stretch of linear DNA with 6 GAATTC sites. Okay, so this linear DNA has 6 restriction sites. Upon digestion with echo R1 will give rise to dash fragments. The second question, B, a circular DNA with the same 6 GAATTC sites upon digestion with echo R1 will give rise to dash fragments. So here we are going to calculate how many fragments will be formed by echo R1 enzyme, the linear DNA molecule, also in circular DNA molecule. Okay, so the formula to calculate this number of fragments formed for linear DNA and circular DNA are, so for circular DNA, if you have n number of restriction site, then you will get n number of fragments. Okay, so if you have 6 restriction site, then you will get 6 number of fragments. Okay, and for linear DNA, if you have a number of restriction site n is equal to, it will form n plus 1 number of fragments. Okay, that is, if you have 6 restriction site, then you have 7 number of fragments. So let's see how circular DNA is giving n number of fragments and linear DNA is giving n plus 1 number of fragments. Okay, first let's see an example for circular DNA. The circular DNA has 4 restriction site for echo R1. Okay, and this circular DNA doesn't have any free ends, so it is a closed circular DNA. The circular DNA has 4 restriction site. So this is a first cut A, second cut B, and third cut C, and the fourth cut considered as D. Okay, so number of fragments formed are AB, BC, then CD. Okay, CD, then one more is DA. So number of fragments formed are 1, 2, 3, and 4. Okay, so it has 4 restriction sites, so it formed 4 number of fragments. Okay, so for circular DNA, if it has n number of restriction site, then it will produce n number of fragments. Okay, next let's see for linear DNA. This linear DNA also having 4 restriction sites for echo R1 restriction enzyme. So consider this is your linear DNA. So this linear DNA has 2 free ends. Okay, so here 1 and it has 2 free ends. Okay, so we are going to digest this linear DNA with echo R1 restriction enzyme, and this linear DNA has 4 restriction sites. So the cut will be 1, 2, then third cut is C, and fourth cut is D. Okay, so the number of fragments formed 1A, AB, and BC, then CD, then D2. Okay, so number of fragments are 1, 2, 3, 4, 5. Okay, here your number of restriction site is 1, 2, 3, 4. For 4 restriction site, you got 5 fragments. Okay, so for linear DNA, for n number of restriction site, you will get n plus 1 number of fragments because it is having 2 free ends. Whereas in circular DNA, you doesn't have any free ends, so it will form 4 fragments for 4 restriction site. Okay, so come to the problem. If a linear DNA with 6 GAATTC sites upon digestion with echo R1 will give rise to. So for linear DNA, it will form n plus 1 number of fragments. So for 6 sites, you will get 7 fragments. Okay, and for circular DNA, if it has same 6 restriction site, so the enzyme will give n number of fragments. So for 6 sites, 6 fragments. Okay, so it's very simple problem if you know this concept. Thank you for watching my videos. If you like my videos, please do subscribe and share.